All right, in this section of our course, we're considering genetics, and I wanted to create several brief videos that outline some of the most important concepts regarding genetics. In this video, we'll go over the difference and relationships between phenotype and genotype. So we'll look at those terms really briefly, and then we'll have some other videos going over other important topics related to genetics. All right, so for these terms, let's start by defining phenotype. So phenotype is a word we use to define the physical appearance of an organism. All right, so phenotype is the physical appearance of the organism. I think it's actually helpful to use that, the similarity in the sound of those two words to help remember. So again, phenotype is the physical appearance. Okay, so throughout this genetics section, you'll be reading and learning a lot about Mendel and what experiments Mendel did to learn about genetics. He really set the groundwork for our modern understanding of genetics. So in this image, I'm showing you a bunch of the phenotypes that he looked at in his experiments, the physical appearances of the organisms that he worked on to help understand genetics. So he worked on this pea plant. He grew a whole bunch of these pea plants in his greenhouse at the monastery where he was living. And he was looking at the different physical appearances. And so he found 7 different physical appearances that he worked on. So quickly, the different physical appearances related to the seed. So here we're looking at the seed. And he either had these gray round seeds or these white wrinkled seeds. So round versus wrinkled was one of the main traits he looked at. He also considered the color of the seeds. The seeds could be either yellow or green, right? So the physical appearance of the seed was yellow or green. In some of his plants, he considered the color of the flowers. The flowers in pea plants are either white or purple, violet. He could look at the seed pod. So he'd look at the pea plant seed pod. Sometimes it would be full in shape. Other times it would be wrinkled in shape. Sometimes the seed pod would be yellow. Sometimes it would be green. The location of flowers could be different. Sometimes they were at the ends of, on the interior of branches, where sometimes they were at the end. And the size of the plant could be different. Sometimes the plant would be very tall, and other times it would be very short. So Mendel looked at these 7 different physical appearances of his pea plants and then investigated the genetics of these physical traits, how the traits in offspring related to the traits in their parents. Okay, so all of these represents different ways that the plant looks, its physical appearance, and that's the phenotype. So let's now consider the other term, genotype. So the genotype describes the genes an organism contains. So phenotype is physical appearance. Genotype is the genes inside the organism. So let's think about the genes and what would be in there really quickly. You'll remember that we've talked about diploid cells before. All of our somatic cells are diploid. Most of the organisms we're going to talk about throughout this course have diploid cells. Remember that when a cell is diploid, that means that it contains 2 of each kind of chromosome. So I've got a picture over here of a diploid situation where there are 3 different kinds of chromosomes: small, yellow, medium, red, large, blue. Okay. And there are 2 of each of them. So we have 2 copies of each chromosome. So as a reminder, chromosomes carry genes. So on these chromosomes are a bunch of genes. Each chromosome probably has 1,000 or more genes on it. Okay. So if we have 2 copies of that chromosome, by definition, we have 2 copies of each gene. So in diploid cells, we have 2 of each gene. So when we're describing the genotype, we're going to think about what kinds of copies of each gene do we have. So there's 2 of them, and we'll think about, are they the same or not? So I'm going to add some symbols to my picture over here. And on the red chromosome, going to say the red chromosome, one of the genes on the red chromosome is gene A. And so here I can put some symbols in the spot where gene A is, and we'll talk about the meaning of these symbols on the next slide. So the first chromosome will have a bunch of genes. Gene A is one of them. The second chromosome will also have some genes. Maybe gene B is one of the genes on this second chromosome. So I put two letters here to represent where gene B is on that chromosome. But you'll notice now the letters are different. There's an uppercase and a lowercase. So again, we'll consider that difference on the next slide. And then again, the last chromosome has a bunch of genes on it. One of those might be gene D. And so here I've placed D on the chromosome where gene D is. And again, I've used the letters to represent the gene. Okay. So when we talk about genotype, we need to understand another term, which is allele. Alleles are just multiple versions of a gene. So each gene can have more than one allele or form of that gene, version of that gene. Each allele of the gene might function differently. And so when we're thinking about the genotype, what genes do we contain? We're thinking about which alleles do we have for each gene. That's what we're really looking at with the genotype. So the different alleles are represented by different symbols. So for example, gene B can have two different alleles, and we might represent it as an uppercase B for one allele and a lowercase b for a different allele. Again, they might function differently. So that's what I had put on these pictures, the picture over here. For the middle chromosome that has gene B on it, I've indicated that the two copies of the gene are consist of different alleles. So one has the uppercase B allele, and the other has the lowercase b allele. Whereas for the gene A, I've shown that there are two of the same. Both of my copies are the uppercase A allele. For gene D over here, I've shown that both of the copies are the lowercase d allele. So here we're building towards ways to describe the genotype. Again, it's what genes do we have? And when we ask what genes do we have, we're really saying which alleles of each gene do we have? Okay. So we'll use these symbols to describe the genotype. And I want to point out one important factor before we move on, that it's these combination of alleles, our genotype, that determine how the gene will function, which will end up controlling our phenotype. So there's a really important relationship between genotype and phenotype. We'll look at that again in just a second. All right, so we've talked about alleles and the different forms of a gene. So let's wrap up by considering the kinds of genotypes we can make out of that information. So there's two kinds. There's going to be homozygous genotypes. I'll give you some examples and a definition in just a second. And there will be another kind, the heterozygous genotype. So let's add a definition and some examples for each of these. So the homozygous genotype is any time that you have two of the same alleles. Again, on the last slide, I was using the letter A, for example. If I have two uppercase A's, two of the same, that's a homozygous genotype. Here's some other examples. Again, two uppercase A's, that's homozygous. We'll look at some situations where the annotation is different, and we have this one allele, which is an I with an uppercase B next to it. We've got two versions of this allele, two copies of that same allele. So two of the same allele, that's homozygous. Here we've got two lowercase d's. That's telling me it's homozygous again. Here's another different annotation, an X with this H beside it. We've got two of those. So again, two of the same, that's homozygous. Sometimes you'll see genes where it's A with a little number next to it. So here, A2, we've got two of the A2 alleles, that's homozygous. All right, so homozygous is two of the same. Heterozygous is just when we have two different alleles. So let's look at some examples of those. So instead of having two uppercase A's, if we had an uppercase A and a lowercase a, that's heterozygous. Here's an example using the I with a letter next to it. I've got an I with an A and an I with a B, two different alleles. So that's heterozygous. Again, uppercase D, lowercase d, heterozygous. Here's this different annotation where I'm putting letters next to an X. This X has an uppercase H, the other X has a lowercase h. So again, two different alleles, that's heterozygous. And then a last example, again, an A with a number next to it, A1 and then A2, two different alleles, that's heterozygous. So we're going to use those terms a lot throughout our discussion of genetics. Homozygous and heterozygous are terms that describe the genotype. And then depending on the question, you'll write down allele combinations like the ones I've shown here, but it'll be different for every specific genetics question that we address. All right, so let's wrap up phenotype and genotype. So to summarize, the genotype are the genes that are in our cells, right? And we just talked about the combinations of genes that we can have. Two of the same allele, homozygous, two different alleles, heterozygous. And those genes control how our cells function. Inevitably, they lead to the phenotype, which again is the physical appearance. So genotype is the genes, phenotype is the physical appearance. And if you're going to remember one, the most important thing is that the genotype controls the phenotype. So organisms look the way they look because of the genes they have. And if we can understand how the genotype is related to the phenotype, we can answer any question we need to about genetics. So we'll stop there. I'll prepare some other videos for you on some other small subjects related to genetics, and we'll hopefully get to the point where we can answer all the genetics questions we want to. As always, let me know if you have questions, and I will talk to you soon.